Good morning. This is Chris Hwang. So the outline of my talk today is I'm going to start off with talking about what are the key drivers for innovation in this area. I'd like to introduce our HiCB platform 2023, what is our development strategy and progress to date, and then it also introduce a T-block, our facility design, which is a critical part of our biomanufacturing platform, and then I'll summarize at the end. First, a brief introduction of Transcenta. We are a global clinical stage biopharma company, and our mission is to employ cutting-edge technologies and innovation to provide patients with effective and differentiated and affordable drugs. Although we do have a rich pipeline of biologics in the area of oncology, bone disease, and nephrology, we do have a thriving CDMO business that provides fully integrated DNA to drug product services for our clients. The CMC development and GMP facility is located in Hangzhou, China, and the work that I'm about to talk about today all came from this site. So what are the industry challenges that's driving innovation in biomanufacturing? Well, certainly biologic has been very effective in treating diseases over the past decades. However, due to the high cost of these treatments, there's been limited access to the patients. Consequently, there's a significant increase in drug pricing pressure, especially in the developing countries. To give you an example, for drugs coming from US to China, typically the price of the drug would decrease by 40 to 90%. And also for domestic drugs in China, after this launch, typically the drug price need to reduce significantly before it can be listed onto the national reimbursement drug list. So consequently, cost of goods or the cost of manufacturing drugs is now a pretty big portion of the drug pricing, such that lowering the cost of goods is not just a nice to have or to give us competitive advantage, but in some cases it becomes very much a necessity. In addition, due to increasing competition, especially with biosimilars, biobetter, or even the new modality in cell gene therapy, the price of the drug pressure becomes even greater. In addition, due to the risky nature of drug development and also with increasing competition, having the right capacity at the right time with a low price becomes even more challenging. With the increase of more complex molecules that are less stable and harder to make, and as evident recently with the COVID-19 need for fast response, industry has been really focused on developing more advanced biomanufacturing platform to decrease cost, increase speed, increase flexibility, increase quality, and decrease risk. At Transcenta, we see the need to develop these new platforms. So in 2018, we started down this journey so that we can develop this more advanced platform we call HiCB. So for us to become staying competitive, to ensure the drug supply, more importantly, also preserve capital so we can invest in R&D, and then we can stay true to our mission to expand patient access. So we envisioned back 5 to 6 years ago that we need to come up with a platform that I would call small and nimble and output much larger. Small and nimble because that provides us the speed and flexibility, but once you have the output much larger, you can significantly increase the output that will significantly lower the cost of goods. So these are some of the key attributes of this platform. It needs to be highly productive so you can maximize the output. It has a fast cycle time with the minimum downtime, typically involve 24/7 operation. It needs to be in smaller scale and footprint that would maximize our space, time, and yield. In addition, this platform will be highly automated, leverage a single use, very simple facility design that will be low cost, very fast to build, to get ready, and to expand and then to replicate. And then we would expand the capacity by scaling out versus as opposed to scale up with minimum regulatory risks. It also will enhance process control and also can produce all proteins, not just a stable protein, but also less stable proteins. And more importantly, it's fast to develop with minimum or no scale up from first in human or phase 1. That's why we feel that the intensified continuous connected processing is a key enabler for us to develop this platform. Essentially, by intensifying the productivity of each unit operations, and then you connected them and remove a lot of the non-value added steps, we get to this very small footprint, highly productive platform that we can put into a flexible and modular facility. So we envisioned that platform was going to look like this back in 2018, that we will have an upstream process that's integrated with capture, followed by variant activation, and then implementing a connected and automated flow-through polishing system that I'll describe more detail later, and followed by UFDF. And the design principle is such that we wanted to maximize the output by continuous perfusion, and then you debottleneck the process by integrating this hybrid continuous downstream process, and then implement cell culture media concentrates and buffer stock blending technologies to debottleneck and to maximize the plant output and space-time yield. And that we will leverage using single-use technology, and in our facility design we call T-block, to really increase the flexibility and speed while minimize the capital investment, and then expand capacity as needed. So this 5-year plan was started back in 2018, and we need to have a platform ready in 2023, which is pretty soon, so that we can be ready for late phase and commercial production. So why continuous perfusion, as opposed to other intensification strategies such as intensified fed batch, dynamic perfusion, or concentrated fed batch? Well, I believe that continuous perfusion has the highest potential for maximizing productivity by maintaining cell density at a very high level and optimizing the environment for maximum specific production rate. We can really get that high productivity. It also, because of the high productivity, there's also minimum node scale from phase 1. It also allows you to enhance your process control and therefore your product control, and that this platform can be used to produce all proteins, stable or non-stable proteins. The perfusion technology eliminates the need for cell culture clarification. That makes it very easy to integrate with the downstream. And by developing our own media, we can significantly decrease the cost of the media by about tenfold. So by doing this, we have developed two different platforms. One we call plug and play, which is we use for phase 1 to pre-pivotal. Typically, viable cell densities are less than 80 million cells per mil with a lower perfusion rate of 1.5 RV per day. The typical scales are anywhere from 50 to 300 liters, and duration is usually less than 3 weeks. And from top clones to process lot, we can do this in less than 10 weeks. And that gives us typically a DNA candidate to CMC IND package in less than 10 months. Once you get to late stage, then we'll go to the push to high platform, which is for pivotal and commercial, where we have a higher cell density, higher perfusion rate. Typical scale will be 300 liters to 1,000 liters with a duration of typically 4 to 6 weeks. And this process allows us to significantly increase the productivity by an additional 30 to 80%, where I'll show you some examples later. So here's an example of the plug and play platform that we developed internally, showing same cell line in doing fed batch versus perfusion for 15 different cell lines, and each color represents a molecule. And the y-axis shown here is the harvest volumetric production rate, which is how much product you produce on a daily basis. So in the typical fed batch of 5.6 grams per liter, using this cell line as an example, then that's normal. That's about 0.4 grams per liter per day. As you can see that using the continuous perfusion, we were able to achieve much higher productivity than compared to what fed batch can achieve. Typically, on average, about more than 8-fold increase in yearly output. And also what's interesting is that the cell-specific production rate is how much product produced per cell is increased by an average about 50%, which is very, very significant. But then you can take this platform, you can then push to high. So again, you can push to higher cell densities by increasing perfusion rate. So in this case, showing perfusion rate from 1.5 to 2.25 to 2.5 reactor volumes per day. Consequently, you see the DPR increase significantly over time. 60% here and 100% there. So by doing this, we can really push our productivity, much higher. And I just also want to emphasize here that in this case, the processing media is not optimized. We simply just push the cell density higher by increasing perfusion rate. So by doing that, showing some examples here of how much we can push the productivity of these cell lines. I'm showing examples of 4 grams per liter per day, upwards of 6 to 8 grams per liter per day. Again, these are per day basis, so that at this range of productivity, a single 500-liter single-use bioreactor can make more than 600 kilograms of drug substance per year. And again, these are not optimized, and we simply just increase the perfusion rate in cell mass. And then we can extend the run further to further increase in output. Very importantly, we also demonstrated that by using this continuous perfusion process, that we can maintain product quality attributes under the state of control. So in this case, for molecule D, we've shown size exclusion, charge variance, and glycan profile. As you can see, that the profiles are fairly steady, and this is a very important part of the process. Furthermore, we demonstrated we can scale the process of the intensified perfusion process. Here shown the cell densities, typically in this particular process, about 120 million cells per mil. We maintain a high viability. In this case, we showed that the productivity of the 2-liter process lock and the 300-liter GMP runs are fairly consistent. Again, these also show consistent product quality profiles. So by using this 300-liter scale process, we can make about 16 kilograms of drug substance per run, or about 175 kilograms of product per reactor per year. Once we scale this process to a 1,000-liter scale and increase the duration, you can see that you can significantly increase the amount of drug substance you can make per run and also per year as well. So with such high productivity, we need to develop a very productive downstream process so that we can handle whatever the upstream process can make. So these are the key design considerations and strategy of how we come up with our downstream process. One is that the single downstream train need to support multiple highly productive intensified perfusion processes as well as intensified fed batch. We have some of the older processes we use are intensified fed batch, but certainly our CDMO clients also have requested intensified fed batch processes. So this needs to be flexible enough to do both. And the principle is that we wanted to increase the output from each unit of operation as much as possible so that when we connected them together, we can minimize the footprint and also the capital expenditures. We want to make sure that the cycle time is very fast and they allow us to maximize the overall throughput. Certainly robustness is a very key criteria. We also need to maintain bioburden control and minimizing complexity and therefore minimizing the risk for our manufacturing and make it easy to operate. And also this platform needs to be very simple and fast to develop and transfer, and that there will be minimum regulatory risk. And that again, supporting multi-product, and that will further improve the process economics. And very importantly, this needs to be ready by Q1 of next year. So we developed this platform using hyper-continuous. So it's not fully continuous, but highly continuous for the reasons that, again, to meet this criteria, it has to be a hyper-continuous. And also, we keep it simple by automating existing batch operations, right? So that how we develop process is really no different than batch processes, but we gain the benefit of the connected continuous process. And that's how we came up with this architecture that I shown early in the slides. Again, it's integrated capture. For now, it's a batch BI work, but we're certainly looking at technology to connect this to our process. And then connect an automated flow-through polishing system, followed by, at least for now, batch UFDF. Certainly, we can't do this alone, so we partnered with Merck KGaA to co-develop this novel single-use downstream technology, and we signed a multi-year technology collaboration agreement back in June of 2020. So fast forward today, we've made a lot of progress over the past couple of years, and I kind of just want to show you where we are. For the integrated capture, we're using the multi-column capture system from Merck. This is the first Mobius system they've just launched not too long ago, and we're working on testing this equipment, and we anticipate to have this ready by Q1 of next year. And this technology would give us these very important advantages. You can decrease the upfront resin cost by as much as greater than 60%, increase the output per day by more than threefold, and also you'll decrease operational and scale risk because of this, because of the automation involved. On the polishing system, we developed this combo system, and has all the advantage of, again, freeing up more than 40% of the pre-viral downstream capacity, up to 10 times increase in output per day, and again, decrease the upfront resin cost by as much as greater than 60%. And that we have the system in our facility since the beginning of this year, and we're doing a lot of testing, as right now this system is ready for GMP operations. And just show you some data on the combo. Here just showing the wholesale protein residues using the batch process versus using this flow-through polishing system. Looking at day 0, day 1, day 2, you can see the significant decrease in wholesale protein as expected, very comparable to the batch process. Similarly, on the size exclusion purity, we've seen a similar performance at the batch process, except this is done fully automated, such that we can process all the product from the entire perfusion run in less than 30 hours, fully automated. And we demonstrated comparable product quality attributes and yields of batch processing. Furthermore, we can modify the flexware, the single-use components, to further increase the throughput and the decrease the cycle time by at least twofold. So that gives you a sense that this technology is very, very productive, and again, it's going to be able to handle whatever is coming from our upstream process. Now that I introduced to you our HiCB platform and the progress to date, I wanted to shift gear a little bit and talk about our plant design T-block, which is an integral part of this whole biomanufacturing platform. With the high productivity of HiCB and use of single-use technology, and allow us to really scale down our operations and simplify our facility design. But to increase speed and flexibility, we decided early on to partner with GCON in working with them to customize their prefabricated modular clean rooms. And the advantage of that is that you can build your building, your core shell and core structure, while at the same time you're building your clean rooms. And by doing things in parallel, it really decrease the time for construction and also simplify getting the whole manufacturing facility to be ready. So for the reasons, and also by using these prefabricated clean room technology, it really improves our predictability of our project, and again, the speed. And I'll give you an idea of how fast it is in the future slides. And here is a layout of our manufacturing facility. You see these are various pods. These are the GCON pods, starting with the cell expansion. And then there's two 500-liter upstream pods, followed by a downstream pre-viral purification and then post-viral purification. Then there's a solution prep area, equipment prep area. And these are all completed in a short amount of time back in early 2018. And then since then, we have added a new cell culture suite in 2020. These accommodate 2,000-liter bioreactors, and then also a much larger purification suite to increase our capacity. So this highly flexible single-use modular facilities was completed about 2 years from the start of design to completion. It could have been much faster for the reasons that I indicated down here, but we believe that this facility from greenfield to having a ready can be done in 18 months. It took us about 7 months from mechanical completion. This has occurred beginning of 2018 to completing our first GMP batch. The whole cost of facility of this initial facility back in 2018 was less than 45 million. So this is the greenfield facility, including this 5-story building for administrative staff and PD support, including land purchase. So fairly inexpensive. And then since then, we have completed over 50 GMP batches with a success rate of more than 98%. Also, we've shown that it's very fast to add capacity. In fact, we tripled the capacity since 2018. And that this facility using the HiCB platform is capable of producing more than 1.2 metric tons per year. And that we can expand this to 2.4 metric tons per year just by adding additional bioreactors. And again, this platform supports both continuous batch processing. So when I joined Transcenta back in 2016, I envisioned that the future of manufacturing would be small and nimble with output of much larger, as I stated earlier in the presentation. And I'm happy to report that we are almost there. Here I want to show you an illustration of the power of HiCB. If you use the same cell line to match the output of 4 by 1,000 liter bioreactor of the HiCB platform, using conventional batch processes, you need 22 2,000 liter bioreactors. Right, that's huge. Total bioreactor runs per year instead it will be 30 versus 450 2,000 liter runs. The capital costs will be significantly lower for the HiCB platform versus the conventional batch. And also, it takes a lot less people to operate. And consequently, you put it all together, the cost of goods is more than 50% lower compared to conventional batch. In addition, from the basic design of facility readiness, it's much faster for the HiCB platform because of the simplicity involved, and the scale of operation is much smaller than compared to conventional batch. Not only that, the continuous perfusion process can produce also less stable proteins where a conventional batch cannot. And also with the improved environmental control of the continuous processes, you can have enhanced process control in the HiCB platform. So overall, this sort of small and nimble and output much larger facility that we envisioned back in 2016 or so can meet these objectives, right? Which is exactly what we set out to do. That's necessary for us to be competitive and also to be able to expand patient access. Here, I just highlight some additional advantages here. You get significantly less upfront capital and then total capital investments. You provide the speed to getting the facility ready. And because of that, you don't have to build a facility years ahead of the demand so that you minimize your capital investment risks. And the projected saving is, by reduction in cost of goods, using an example of 2 metric ton yearly output, you can save more than $100 million, and that money can plow back into your R&D to develop additional products that's going to eventually serve the patients. So at Transcenta, we decided early on that we would implement the HiCB platform for all our molecules in our pipeline no later than the PPQ commercial stage. And that's one reason why the platform needs to be ready in 2023. But certainly we have products that was developed earlier that are all done using conventional fed batch and batch processes. And then we were just leveraging our capability to bridge the process from the early stage to the late phase and then eventually to the PPQ commercial stage. As you can see here, here's an example where in the molecule A, we converted fed batch to perfusion and then modified downstream, and we were given the approval to proceed from CD and FDA regarding this process change. And likewise, we would do for some of these other products as well, using the same comparability bridging strategy. Some of the later stage molecules, more recent molecules, we start with perfusion right away, followed by batch. And once the continuous connected downstream is ready, then we will implement for the late phase, and again, ready to go at the PPQ commercial stage. And then for any later molecules, it will be the full HiCB platform from the very beginning. And this is our plan, and it's already in progress. In summary, I have shared with you the key industry challenges that are driving innovation in biomanufacturing. And in particularly for Transcenta, with the significant drug pricing pressure and competition, it really makes it necessary for us to implement these highly productive processes starting 2023 to really drive down the cost of goods of our commercial products. I share with you some exciting progress made over the past years around the continuous perfusion platform, where push to high can result in more than 15-fold increase in volumetric productivity compared to fed batch processes, while at the same time improving process control. And that the beauty of the HiCB platform is that it's designed to simplify process development while minimizing the tech transfer, scale, and manufacturing risk, which are very important criteria for us. And that with the development of the highly intensified downstream platform in collaboration with Merck KGaA, that we have shifted bottleneck back to the upstream, and that this technology is ready for GMP operation in Q1 and 23. So together, HiCB and T-Block can provide an output of much larger, slower, and more costly facilities in a small, highly flexible, and lower cost facilities to support multi-product, speed to clinic, speed to market, while again, significantly lower the cost of goods. And that this platform is fully accessible to our partners and CDMO clients. So if you have any questions, please feel free to reach out to us, or if you need our support, feel free to reach out to us as well. Lastly, I want to acknowledge our CMC team in Hangzhou for the fantastic work that they have done over the past years, while at the same time delivering on all the internal and external projects. And also to our collaborators that without their help, that we would not get to where we are today. So with that, I want to thank you for your attention. And again, reach out to us if you have any questions or support needed. And I hope you have a great rest of the conference. Thank you.